How are you all?
Good, thank you.
Good. How are you?
Good. What's the time?
I think it was like 5:30 on the dot.
Okay, so just wait 1 more minute. So if anybody join. So what do you think about this class?
It's very interesting.
It's fun. It's just a lot. That's it.
Yeah, that's a lot. That's
Yeah.
right.
Mm-hmm. It's as I told that it's really easy stuff. You if you read you can understand. But
Yeah.
the thing is, yeah, it's huge. That's the problem. I think I should start from here, right? This slide?
Yeah.
I think so, as much as I remember.
Yes.
People are still joining, so. Okay, let me move this one. I hope. Okay, so. Okay, so um, so last Monday we talked about all the different types of microscope, their principles and those. Okay, just give me a second. Sorry. Okay, so now this part of the chapter actually talks about how uh, you can actually see the specimen under the microscope. Okay, so microscope, all the microscope, they all different types of microscope, they work differently. We now we know how to use light, properties of light differently. Okay, but this part of the chapter actually will talk how we have to make specimen for different types of microscope. Okay, and so that we can see the specimen properly, or you can say we can see the specimen in detail. Okay, so basically this part of the chapter is about uh, the making the smear on the slide, then how we give the color. Okay, and then how we see the specimen. So as I told that we use different properties of light. Okay, so we use light differently uh, for each and every cases of microscope, every different types of microscope. Okay, for each different types of microscope, there is a special type of uh, specimen preparation. Okay, like that day I told, like the light microscope, that the microscope we use, um, in the lab, we can use both the living specimen, like if you want to see the ladybug, okay, ladybug is moving, really large specimen, you can see that ladybug with your open eyes, but if you want to see in detail, okay, all the hair structure of ladybug, what you can do, you can use the light microscope, okay, because light microscope, the principle of light microscope is it will allow you to see the natural color, and you can see the living specimen, okay. On the other hand, by using the same microscope, that light microscope, you can even use the dead bacterial cell, okay. So basically this microscope is for both the living and non-living organism. You can see small particles, small organisms, very small organisms, or you can see a pretty large living organism, okay. If you want, you can give the color to the organism. If you don't want, it's okay. Now, for let's say that phase contrast microscope, okay? Phase contrast microscope, if you want to give the color, or if you have to give the color the specimen, then you cannot use the phase contrast microscope, okay? Because phase contrast microscope, the principle, basically it works in such a way, you really do not need to add any color, okay. Color, all the dyes, it is actually type of salts, okay, and inside all these dyes there, it's called chromatophore, that actually reflects light. It helps to see the light better. Anyway, so the phase contrast, it's not for the colored specimen, but if you really want to see the natural color, then phase contrast work even better than the light microscope. On the other hand, when it is an electron microscope, the magnification is really high, 1,000 times higher, 10,000 times actually higher than the light microscope. So you can see even the detailed DNA structure, but you cannot see colored stained specimen. You cannot see living specimen. It has to be dead specimen. Not only that, the thickness, it's so the specimen has to be so thin actually, to make that specimen, to prepare basically for the electron microscope, we have separate machine, okay, and we use that machine to make the specimen section. Then only we can use that thin section under that electron microscope, okay. So basically what I want to say is that for each microscope, we really need to make specimen ready so that we can use the principle of light and the principle of that microscope properly, okay. So this part of this chapter, we are actually going to talk mostly about that. We only work over here mostly for light microscope, bright field microscope, okay. Usually what we use in our lab. So first thing to do or to see the specimen under a microscope is you have to take the microscope, take the specimen under the microscope, okay. For that, what we usually do, we actually use a small thin glass slide, okay, and we keep the specimen, or we took the specimen on top of that slide. And this process is called that mounting a sample on the slide. So basically when we take a specimen on a glass slide, and that is called the mounting of the sample on a slide. Now, how the slide is prepared, basically how we mount the specimen on the slide, that depends on what type of specimen it is. Is it living specimen or preserved or dead specimen? Okay, that's one character we need to look for. Then second is that actually what you want to see. Okay, do you want to see the cell surface? Do you want to see just the outside? How much detail basically you want to see? And the third is the type of microscopy available. What type of microscopy? The light microscopy, or what type of light microscopy you have. So these are the three conditions. Depend on that, we actually mount the specimen. Okay. Now, mounting, there are two different mount, wet mount and fixed the specimen. When it is wet mount, it could be the dead sample or maybe the living sample. It could be both when it is wet mount. When it is fixed mount, that means it's actually dead sample. Okay. To fix the specimen onto a slide, it requires the specimen to kill. So, okay. So the wet mount, first one, wet mount, what you can do, like you can grab a slide, okay? You just take a slide, let's say this is the slide, and then take the specimen over here at the center mostly, and then take a drop of water. If it is a solid specimen, then you have to take water. If it is urine sample, you really do not need to add any water. Just take a drop of urine sample. Or so basically, if it is liquid sample, just add one drop of sample. If it is a solid sample, then you have to add one drop of water so that that light actually can go through the water. Okay, different media, and that actually helps the image to be, it actually creates a much brighter image. So that's the wet mount. Now wet mount, let's say you just imagine, like as I told, the ladybug. Okay, so ladybug, so you are actually looking for a living specimen. So just imagine that this is a slide and this is the ladybug. Okay, and you are trying to focus your microscope. What will happen? After a certain period of time, you will see that there is no specimen, because it has locomotory organ, right? So it will move away from the slide. So that's a problem with this type of slide that we usually use in the lab. So for moving and for the large specimen, basically for the living and for the large specimen, we have a special type of slide. It is called hanging drop mount. Over here, what happened? It's a slide, but the center, it is, this part is actually depressed. Okay, so this part is looks like a bowl. Okay, it's a depression. So what happened? So you take water and you take specimen over here, so that the living creatures, they actually cannot walk away of the slide. So that's the hanging drop mount. Usually most of the time we use for the living and the large specimen. Otherwise, if it is a small specimen or dead specimen, this flat slide is perfect and to do the wet mount. So from the name, this wet mount, both in the plain slide and inside a hanging drop mount, you can understand that we use water or any liquid solution. So that's why it's called wet mount. And as I told, by wet mount, you can do both living specimen, okay, and you can see dead specimen. So both type of specimen can be seen by doing wet mount. Now the fixing, when it is fixing, then it's only about dead cells, okay, or dead specimen. Now, when it is fixing means we are going to fix something. So what we do, we actually take the slide and we fix the specimen over here. Means we spread the specimen in such a way so that the specimen gets stuck with the slide. Okay, so one purpose is fixing is to kill the specimen. That's number one purpose. Number two purpose is to adhere the specimen tightly onto that slide. Okay, so these are the two things. That's our target for the fixing. So what we do, we take a slide and we spread a thin film made from a liquid suspension of cells and air drying it. So basically what we do, we take the slide and most of the time, as I told, that it is the dead specimen, and fixing, usually we fix very small and minute specimen. So what we do, we have to make a really thin spread of the specimen onto that slide. And when we spread, we try to spread the specimen evenly. Basically, we want to make a thin layer of specimen onto that slide. And that thin layer of specimen is called smear. Okay, so when we spread the specimen onto the slide with the help of water, it's called smear. You can understand that if it is a dry bacteria, okay, maybe 100 bacteria are there, but it's a dry specimen. So it would be very difficult to spread that dry specimen evenly to make a thin layer on the slide. So what we do, we actually add a little bit of water, okay, on top of that specimen, and then it becomes very easier. It becomes smooth and easier. It's like a paste, and we actually can make a thin layer of specimen. Okay, so, and that is called smear. So the smear preparation or learn how to make the smear, it takes time. Okay, so in this lab you are going to learn that how to make a thin smear. If your smear is good, bacterial smear actually, if your smear is good, then it takes really nice color. If smear is really thick, it would be very difficult to identify the bacterial cell. You cannot see that separate bacterial cell. Basically, all the bacteria will be on top of each other. So you really cannot identify the shape of the shape and arrangement of the bacteria. Okay, so making a thin layer, it's very, very important. So now when we make the smear, what happened, as I told, that we add a little bit of water and then we spread it. Okay, and that way actually we try to fix the specimen onto the slide. Sometimes instead of water, we even use chemicals. If we use chemicals, that means by adding that chemical, we actually kill the specimen. Okay. Now, over here, you can understand that slide and then water or maybe liquid chemicals and the specimen. So it's wet, right? So if you want to add any color, if you want to stain the specimen, if it is really wet, then it will drip from the slide, and you really cannot, and that there will be a different opacity and light will go so many different directions, you really cannot see the specimen. So what we do, first we make a thin smear of specimen onto that slide, then we actually allow the slide to be dry, allow the specimen to be dry, okay, in the air, and this process is called air drying. Okay, then once the air drying is done, that this step is very, very important, that air drying part. When air drying is done, then what we do, we hold the slide in front of any Bunsen burner, or in this lab we use micro incinerator. So in front of any burner, we actually hold the slide and we heat it up very little bit up. Okay, our intention is to kill the bacteria, that bacteria or any specimen, and other thing is so that the specimen actually get tightly attached on the slide. So these are the two purposes, and that is called heat fixing. So when we are talking about the fixing, there are two things. First, actually, when it is fixing, first step is actually making smear. So it's already a sample or specimen is on the slide. Then what we do, then we actually air dry. And then number 3, we do heat fixing. Okay, means we actually add little bit of heat, and as a result, we make it secure that all the specimens are tightly attached on the slide, and this whole process is called fixing. Now, when in this picture, actually, how we do, heat fix, that's what this slide is talking about. So heat fixing, we have hot plate like this one in this picture. You can just keep the slide on hot plate. That actually works. Basically, we provide little amount of temperature heat. And this is the micro incinerator. In this lab, we usually use micro incinerator. We just hold the slide in front of the micro incinerator for 2-3 seconds, and the specimen get fixed onto the slide. We have one more ways. Sometimes we use so many different type of chemicals, you can say, the formalin, acetic acid, so many different types of chemicals are there based on what type of tissues, what type of cell you are going to see. Based on that, we actually can do chemical fixing. If it is chemical, then we really do not need to use any heat fixing, or we do air dry. So we basically treat the specimen with the chemicals and let it dry, and then from there we can move. Then the next step is staining. So staining means we actually give the color, we add color. Now, what's the purpose of the color? Now, just imagine, let's say that this is VSAB, Bacillus subtilis, okay? And Bacillus subtilis, it's a rod-shaped bacteria, and they actually make a long chain. If you give color, then it becomes really clear, the shape, size, everything. Okay, if you like, what happened, like this part of the cell, how it will reflect is little different than this part of the cell, how is going to reflect light. So and because all these cells, we are actually giving color, so inside the color, as I told, that stain all are salts, and it's called chromatophores. So different chromatophores absorb different frequency of light. Okay, they absorb different frequency of light, and they reflect that light differently. So that's the reason, if we give color, so as I told, that reflection from this area and reflection from this area would be completely different, and you can see the specimen clearly. Okay, so that's the purpose of giving the color. There is another purpose to identify, like not all specimen take all type of dyes, okay, based on the ionic composition of the specimen. They absorb and they reflect different types of stains. You can see like gram-positive bacteria, they absorb they actually show the purple color. Gram-negative bacteria, they are pink because of their cell wall structure, the ionic composition of the structure, chemical composition of their structure. So these are the purpose of giving color, or you can say to stain the specimen, basically to make it clear. I just told you the science behind this, how actually the color actually makes difference. So stain, they are colored organic compounds used for staining microorganisms. Certain salts made up of a positive ion and a negative ion, so contain salts. So these stains, they are actually made up with salt. Whenever it is salt, each salt has a positive component, has negative component. Like let's say sodium chloride. So this is the sodium, this part is the positive component, this part is the negative component. So all salts are like this. Basic dyes, these dyes have positive charge. So in this dye, the positive ion, number of positive ions are more than the number of negative ions. Okay, so you can understand that if it is a basic dye, let's say over here we have 5 sodium but only 1 chlorine ion. Okay, so basically that dye make the specimen positively charged over here in this case. So if it is a basic dye, so if the dye is positive, and we know that always the opposite ionic charge attract each other, right? So if the specimen is negatively charged, then if we add another negatively charged stain, then it will not take, the specimen will not take the negatively charged stain. But if we add in any positively charged stain, okay, then it will stick onto that specimen. Okay, so that's the science behind. So basic dye, they have positive charge. Okay, so they easily can bind to the negatively charged molecules. All the bacterial cell wall, you will see that it has phosphate PO4 -3. So that phosphate, its bacterial cell wall is mostly negative. Okay, so they easily grab this basic dye. And that's the reason in this lab we are going to use all the basic dyes like crystal violet, methylene blue, saffronin, basic fuchsin, all are basic dyes. Means these are all positively charged. On the other hand, acidic dye, they are negatively charged. Nigrosin, picric acid, eosin acid, India-link, all are actually acidic dye. Now, acidic dye, it's negatively charged. So mostly when the specimens are positively charged, like there are few protozoa, large, helminths, nematodes, we use those acidic dyes. Now there are 2 different types of, so these are all about the staining, okay, and this basic dye and acidic dye, those are the staining material. Now the process, 2 different ways are there: positive staining and negative staining, like how we give the color. Okay, now positive staining over here you can see actually by this process we give the color directly to the specimen. Okay, we really do not stain the background, we only stain the specimen. That's the positive staining. Negative staining, we don't touch the specimen. Instead, we actually color the background. Okay, these are the 2 different ways, and that's the difference between positive staining and the negative staining. So I hope you don't mix it up. Okay, when we are talking about basic dye, acidic dye, we are talking about the color, about the stain. when we are talking about positive staining and negative staining, we are talking about the process, okay? By positive staining, we give the color to the sample, to the specimen. By negative staining, we don't touch the specimen. Instead, we color the background. You can see. If we really want to see the shape, like let's say the capsule, okay? Capsule means if it is a prokaryotic cell, outside of the cell there is a slimy layer, thin slimy layer, okay? Slime layer actually. And it's a protective layer for, especially for the bacteria, and bacterial pathogenicity depends on that. So this is the capsule. Capsule, this color, this slimy layer, it's very difficult to give the color to the capsule, okay? And to identify the pathogenicity of bacteria, as I told that this capsule is responsible for the pathogenicity of the bacteria, it is very important to identify that if bacteria has the capsule or no. So what we do usually most of the time to identify that bacteria has capsule or no, we actually do the negative staining, okay? That gives a clear shape and structure, and that capsule is slimy layer, and in between the outer cell wall there is a little gap in between capsule and cell wall. So by doing this negative staining, we can actually identify the presence or absence of the capsule. Negative staining, the Nigrosin India link, we frequently use Nigrosin and India link. Over here, we really don't do any heat fixing. So most of the time to observe or to study the characteristic features of capsule and spore, we do negative staining. These are the different types of staining and different types of bacteria, you see. Now, staining, there are two different types of staining, simple stain and differential stain. Okay, so again, we are into the process, okay? Over there, that slide, we talked about positive and negative staining, means, we are giving specimen the color or we are giving the background the color. So that's the positive staining and negative staining. Over here, two ways to process, simple and differential staining. Like how many stain actually we are going to use over here. Based on that, we have simple staining process and differential staining process. Simple staining process means we just want to use one color. Just one color. You only want to observe the shape. You want to identify, like if I ask you, okay, what shape of the bacteria is, okay? Or you want to identify, like let's say it's a diarrhea patient, okay? So diarrhea possibility, either it is a protozoa, round-shaped, or it's E. coli. E. coli is rod-shaped bacteria. So you really do not have enough time, okay? The patient is going to die. If you delay, if you want to culture the specimen, if you take 36 hours, 24 hours to culture the specimen, patient will die. You have to give medicine immediately, okay? Either antiprotozoal or antibacterial, okay? So only thing you need to identify the shape, you just do the simple stain, okay? Just give one color, and oh, it's round, that means it's protozoa. It's rod-shaped, that means it's E. coli, okay? So that's the simple stain, and that's when we use simple stain. You want to see the size, okay? You want to differentiate the size between these two organisms. You really do not need to do lots of complicated process, okay? Complicated steps. Just give one simple color and you can check what you want to say. So that's the simple stain, means we just use one, just single dye, okay? You can use the crystal violet, it will give you purple color. Okay, basic food stain, it will give you pink color. Saffronin, it will give you pink color. So that's a simple stain. Differential stain, it's little complicated. We use more than one stain, like a gram stain. Okay, what we do at the beginning, we give that crystal violet. We treat the bacteria with crystal violet. Some bacteria, if it is gram-positive bacteria, they take that, their cell wall is so thick that they take that crystal violet. So they will be purple in color. But the gram-negative bacteria, they really have very, very thin cell wall. So they don't take that crystal violet. Okay, it does not goes, what do you say, inside the cell wall in case of gram-negative bacteria. In that case, what we do at the end, we use that bacteria with saffronin. Okay, so saffronin, it gives that gram-negative bacteria that pink color. So basically over here, what we want to do, we are actually examining the structural differences between the two groups of bacteria. We are going to see if this bacteria has thick cell wall or it is a very thin cell wall. And that's the difference between gram-positive and gram-negative bacteria. Gram-positive bacteria, they have a very thick cell wall, very thick. Gram-negative bacteria, they have very thin cell wall. Okay, and we want to see that. So antibiotic, for gram-positive, a different type of antibiotic. Gram-negative, it's completely different. Okay, we have broad spectrum antibiotics. Sometimes we frequently, actually not sometimes, we can use that broad spectrum for both gram-positive and gram-negative. But by giving that, you are going to actually harm the patient. Okay, so if you really want to do properly treat this patient, then you have to give the proper medicine. For that, you have to identify that bacteria. It is a gram-positive bacteria. Okay, so I can give this antibiotic. It is gram-negative bacteria, then I have to give this antibiotic. To do that, by doing the differential staining means you are treating the bacteria, group of bacteria with more than one stain, more than one dyes, and that's how we can identify that which group the bacteria belongs to. Okay, so that's the difference between simple stain and differential stains. Over here, gram staining procedure. Okay, these are the steps. At the first step, we actually use the crystal violet. That's our primary stain. Primary stain, the definition of primary stain is the stain that we use at first. So that's the primary stain. At the beginning, the first stain of the process is called the primary stains. So when we treat the specimen with crystal violet, or you can say the primary stain, all the cells or all the bacteria will be purple in color. Then what we do, we actually rinse the extra stain with water. And then in second stage, we use iodine. Iodine, that's the mordant. What is mordant? Mordant actually help primary stain to go deeper into the cell wall. Okay, so mordant, let me write it down, very, very important, helps primary stain to go deeper into the cell wall. So basically, mordant in any staining procedure, not only the gram stain, in any staining procedure, that gram stain help primary stain to sit inside the cell wall. Okay, so that's our second step. Third step, we use alcohol. Alcohol, it's decolorizer. Decolorizer, what it does, it washes away stain. Extra stain, any extra stain that is not tightly sitting inside the cell wall, it will be washed away by using the alcohol. Okay, so that's the decolorizer. Now what happened at this moment, both gram-positive bacteria and gram-negative bacteria will look purple. Okay, they actually took that crystal violet, and then iodine pushed the crystal violet, so all are purple. But as soon as you use alcohol decolorizer, remember I told that gram-negative bacteria, they have a very thin cell wall. Okay, so thin that this alcohol actually clean away or washed away all the crystal violet from them. At this moment, all the gram-positive bacteria, they will be purple, but the gram-negative bacteria, no color, because they actually lost all the crystal violet color, because they have a very just 1-2 thin layer of cell wall. Okay, and iodine cannot actually help in that case primary stain, and whatever goes in, alcohol actually rinses away all the color. So after this step, gram-positive bacteria, they are purple. Gram-negative bacteria, they do not have any color. Last step is the safranin. You can see that this is the second dye we are using. So first we used, that's our primary stain. First stain, that's the crystal violet. Now this is our, either we can call it secondary stain. Okay, either it's secondary or it is counter stain. Okay, so what will happen? Look, in case of gram-positive bacteria, the cell wall is completely filled up with crystal violet. So no place for safranin. So this group of bacteria will be purple. But this group of bacteria, gram-negative bacteria, they really do not have any color. So they are going to take that safranin color. So after these steps, this gram-negative bacteria, they will be pink, but gram-positive bacteria, they are purple. Okay, that's how we can differentiate between these two groups of bacteria. It's very easy way to identify the bacteria. Okay, and we used two different types of stain, like crystal violet and safranin, and that's why this staining process, that gram staining process, is called differential staining process. Exactly same way we have acid-fast stain. We use different, primary stain, we use, instead of crystal violet, we use carbol-fuchsin. Instead of safranin, we use methylene blue. Okay, but we use more than one type of dye. So again, acid-fast staining process is also differential staining process. This is the way we do gram stain, and I really want you to memorize that for the lab. Every day you need it. So crystal violet, we keep it for 1 minute. So it goes like, make a thin smear. So smear, then air dry, and then heat dry, heat fix it, and then crystal violet 1 minute, then we rinse it with water, and then mordant, gram iodine, and we treat the cell 1 minute. We rinse it. After each step, each dye or each chemical, you have to rinse it, excess color or excess stain. After washing, then we treat that with a 95% alcohol. It's called ethanol, and we just go for 10 seconds. Then we rinse it with water. After water, we use safranin for 1 minute. Then again water, and then blot the extra stain and the water, and you are done. So this is the exact process we use in lab to do the gram staining. Acid-fast staining, there are a group of bacteria like Mycobacterium smegmatis. In this lab, you are actually going to work on these bacteria, Mycobacterium smegmatis. These bacteria, another species of Mycobacterium is Mycobacterium tuberculosis. So you can understand that this, specific genus, these bacteria, they are responsible for so many really bad, ugly diseases like tuberculosis. Okay, so we are not going to work on tuberculosis, but we are going to use smegmatis. It's little harmless than the tuberculosis, but it's not, non-pathogenic. So anyway, that Mycobacterium, this group of bacteria, if you observe the cell wall, they have a very thick cell wall. Okay, so outside they have a very thick cell wall, but they have one special type of fat. Okay, it's like oil, kind of butter kind of stuff, and it is called mycolic acid. So you can understand that oil and water, it does not mix, right? You cannot mix oil and water properly. Exactly same way, all these stain like, like saffronin and all, crystal violet, they actually cannot mix up with that mycolic acid. So what we need to do, we actually heat up, we need to add heat, high heat, so that that mycolic acid get dissolved into the solution. So in this case, our primary stain is carbolfutsin, not carbon, carbolfutsin. And while treating the specimen with carbolfutsin, we actually use heat for 2 minutes. Then we use alcohol, 95%. In this case, we actually use acid alcohol. That acid alcohol, so that we can rinse away all the mycolic acid thing. Then our counter stain or secondary stain is methylene blue. Okay. In this case, our positive color means acid fast positive color is pink, and our acid fast negative color is blue. Okay, we can identify this group of bacteria. So they have a thin cell wall. They actually gram-positive bacteria, but they have mycolic acid. So that's why this group of bacteria is called acid-fast bacteria. And to treat or to stain acid, this group of bacteria, gram stain does not work. So that's the reason we use acid-fast stain. Capsule staining, capsule, as I already told, that it really does not take the stain. So usually what we do, instead of positive staining, we actually do the negative staining. We actually stain the background so we can see the shape of the bacteria, size of the bacteria. Over here, different process, different procedure. We really do not do flagella, endospore staining, okay, but we do gram staining and acid fast staining, and I already talked about these three here. Okay, so that's the this chapter we are done. Do you have any question from this chapter? Anybody? So you couldn't hear me at all, people?
We heard you, Miss Nandi.
Sorry, I was on mute. No, I'm clear.
Clear? Oh, I see the chat. Okay, so do you have any question?
No,
No,
ma'am.
ma'am.
No? Okay, so should I move to the next PowerPoint? Next
Yes, ma'am.
chapter?
Yeah.
Chapter 3? Okay, let me share the screen with chapter 3. Where is? Okay. Do you see my screen now?
Yes, yeah, we do.
Chapter 3? Okay. Okay, so this chapter 3, this chapter, basically we are going to work on the cell, okay? Cell structure. But before we go into the prokaryotic cell structure and eukaryotic cell structure, this chapter actually talks a little bit about the historical discoveries
Mhm.
that actually gave the idea of our current understanding. Okay, it actually helps to build our current and our understanding about the microbes. So basically, we are going to work on different historical experiments. Okay, and how those experiments actually helped us to build the current concept on, like microbes, you can tell the cell, different organism. Okay, so and then towards the end we will work on the prokaryotic cell structure, and then the different cell components and cell organelles of both prokaryotic and eukaryotic. So let me start. Okay, so you can understand that the world of microorganism at this moment, it's really diversified, huge varieties, so many different shapes, sizes of organisms and are there. Not only that, their metabolic activities, their characteristic features are also different. Okay, now so now we know that different metabolic activity, like we know gram positive bacteria, gram negative bacteria, but previously our prehistoric people, they really did not have any idea about how they knew about cell, they knew about microbes, they used to live with microbes, they used to use microbe, okay, to ferment food, to make medicines, but they really did not have any idea about how actually microbes originate, or you can say from where actually they are coming. Okay, so they really did not have an idea. Famous philosopher, you can all of you know the name, oh my god, Aristotle, 300, what is happening, why I'm losing my slide. So Aristotle, you can understand that 384 to 322 BC before Christ, so many years ago, Aristotle told that life actually can arise from non-living matter. As I told, that they really did not have any idea that how, from where all these cells or all these microbes, they are coming from. Okay.
Miss Nandi, what does your screen look like?
I was about to say the same thing. I'm still looking at PowerPoint chapter 3, PowerPoint chapter 4. It's not on the slide.
Yeah, we don't see
Really?
anything. Yes, ma'am. We just see all of your documents.
You don't see my screen? This screen?
Well, we can see your screen, but it's not like the last PowerPoint. We don't see your slides.
Okay, let me.
We see documents.
Yeah.
Yeah, I got you. I'm trying to. I cannot. Why can't I share my slide? Let me see. Yeah, my slide is here, but do you see it now?
Yes.
Okay.
Yes, ma'am.
Okay. Okay. So over here. So basically that Aristotle, the previously, as I told, that prehistoric people, they used to think that cell actually originate spontaneously, or maybe it's a cards, or there is a vibe, okay, or there is a heat. Because of the heat or because of the vibe, microbes can originate or cell can originate. So many different concepts were there. Okay, one of the famous concept that went for a long time, that was spontaneous generation theory. Okay, Aristotle, Greek philosopher, he actually started that spontaneous generation theory. He proposed that spontaneous generation theory. He told that cell can arise from any non-living matter. If there is a vibe or heat, okay, cell can arise. And he claimed that he noticed that on a road, like frog originates suddenly from the vibe. That's what he claimed, that he actually saw. He noticed that several times. So that this spontaneous generation theory, it went for a long time. Like they at that moment, they know the presence of microbes, but they really do not have any idea about their origin. One of the scientists, John Needham, so basically the people were, at that moment, so from here, from John Needham, we are talking about, after microscope, okay? So we have seen the cell, both the plant cell and animal cell under the microscope. So now we know about the existence of microbes and existence of cell. But we are struggling with the origin of cell. Okay, so all the scientists are, lots of scientists are actually behind that thing, that they were trying to find out how actually cell arrives or originates. So one of the scientists, John Needham, what he did, he was the supporter of spontaneous generation theory. And remember one thing, that as I told, that spontaneous generation theory actually was proposed by Aristotle. That time we really did not have any microscopes, so long time ago. So after so many years, John Needham, he was actually a famous supporter of spontaneous generation theory. So what he did, he did an experiment. By the way, this chapter is full, lots of experiments are there. You really need to memorize all the experiment and who did what type of experiment, that you have to remember. Okay, so one of the experiment, John Needham's experiment, what he did, he took jars. I don't know, I cannot write it down now. I cannot use my pen. Then how I am going to, what happened? Okay, now I can write. Okay, so John Needham, what he did, he took jars, okay, two jars, and he took broth. Broth actually liquid nutrient media, and inside the broth we have all the supply of nutrients and special elements required for any cell to grow, you can say. So that's the broth. So what he did, he took broth, okay, and then so basically he took broth means he is providing the food supply to the microbes. So then he boiled that broth, okay, and he sealed, after boiling, he sealed the jar. He made it airtight and he kept it. After a few days, he noticed that broth became cloudy. Cloudy means basically lots of cell actually started dividing and started growing. And that's the reason that broth became cloudy. So he claimed, look, he broiled the broth, means if inside the broth, if there is any living cell, he killed by boiling that broth. Okay, that's what he claimed. Number two, it was airtight. Okay, he didn't allow air to go in. Okay, so the microbes couldn't come in from the air, but still inside that broth, new cells arose. And by this experiment, he claims that he actually supported that spontaneous generation theory. Now, Lazzaro Spallanzani, almost same time, after a few years, who did the same experiment. But what he did this time, he actually boiled the broth for a long period of time. So he increased the time, and exact same experiment of Needham, but only thing he did, he extended the time. Okay, he boiled the broth little bit more time. So and then he sealed the broth and he kept it, and broth was completely transparent and clean for a long period of time. There was no cloudiness. And from this he claimed that John Needham, he did the experiment, he was going towards the same, right directions, but the thing is he boiled the broth, but the time for how long he actually boiled, that time was not enough, sufficient to kill all the microbes or all the germs inside the broth. So he denied, Lazzaro Spallanzani, he denied that spontaneous generation theory. Okay, and he disproved John Needham's experiment. Then Francesco Redi, as I told, that lots of scientists were actually trying to disprove that spontaneous generation theory. So John Needham, Lazzaro Spallanzani, both were working with the broth, okay? But Francesco Redi, he took meat and he actually worked on the maggots of the flies, and ultimately he disproved the spontaneous generation theory. This Redi's experiment, it's very famous experiment. What he did, he actually took 3 jars. Okay, so one jar was, and in each jar he took a piece of meat. Okay, then 2 jars he kept open, 2 jars were tightly sealed. Okay, and 2 jars he covered that container with gauze. Okay, so basically 6 container was there. 2 were open, completely open, so microorganisms can go in, flies can go in all the way up to the meat. They can sit on top of the meat. They can lay eggs, which is called, and from the eggs, larvae can come out, which is the maggots. Okay, over here, it was tightly sealed, so no microorganism could go in. Okay, and over here, 2 container, it was covered with gauze, so the flies, they can come all the way up to the gauze. They can get the smell. Okay, that attracted the smell of the meat, actually attracted the flies. So because of that smell, all the flies came up to this gauze. They were waiting and sitting. While sitting, they lay eggs, and we can see maggots on top of the gauze, not on the meat. So by doing this experiment, he explained, look, that these 2 containers, this container was open, right? So the maggots, previously, the scientists, they told that, look, over here, all these creatures, small maggots, they just originated from the air. non-living meat. That's the non-living material. So from meat, all these maggots actually originated. Okay, that's what other scientists demanded or claimed. But he told these maggots, they are actually the parts of the flies. They came from the flies, who are the living organism. So flies, they sit on top of the meat, they laid eggs, and from the eggs, because the eggs were getting food supply from the meat, so from the eggs, larvae came out. Okay. And they got oxygen, everything they got. Basically, all the supply for the life were there. So from the egg, maggots came out. So all these maggots didn't came out from the meat. It came out from the fly. Over here, flies couldn't reach all the way, even they didn't get any smell. So no flies, no meats. No maggots. Okay, so if maggots originated from the meat, then we could see maggots over here too. But there is no maggots, means there is no relationship between the maggots and the meat. Over here again, look, the flies are here, so we can see the maggots. It's a very famous experiment. From this experiment, he actually disproved the spontaneous generation theory, and he supported the biogenesis. Biogenesis means life only can originated from the life, not from the non-living material. Okay, it's a very famous experiment. Then Louis Pasteur, another great scientist, he did lots of things. So this was the last, experiment. This Louis Pasteur's, it's very famous. It's called Swan Neck experiment. After this experiment, all the scientists agreed that life only can originate from the living organisms, from not non-living organisms. So this is the last experiment by disproving the spontaneous generation theory. So what he did, he took glass flask, okay, and by apply, by applying heat, what he did, he actually give this shape, the neck of the flask. He made it like this, okay, he gave S shaped, and he called it swan necked flask. Now, science behind this shape is it's the pure physics, like when dust particles, okay, air, when they move through this type of shape or tube, air actually can move all the way inside the flask. But any heavy particles heavier than the air, they actually get settled down at the bottom of this area. Okay, they get deposited over here. That's the physics. I really don't want to go into that detail, but that's the science. That anything heavier than the air must be settled down on the bottom of that S neck. So what he did, that's the reason he actually gave that S shape to the neck of the flask. So he took broth, again, same thing, the broth, okay, he boiled, and then he kept that flask for long period of time. Look, the air could go from here to inside the flask, but all the microorganism inside that air, they couldn't go in. So he allowed air to go in, but he didn't allow any microbes, any heavier particles to go in. Second set of experiment, the same swan neck, he took the same broth, he broiled, but what he did, he cut the neck. So there were two flasks, one flask with that swan neck and this one without the neck. What happened after some period of time, he noticed that broth inside the swan neck, there was no contamination. It was really clean, and it was not cloudy at all. But this part, this flask was completely cloudy. The broth inside this flask was cloudy. Why? Because microorganism could come in, okay, with the air, but over here microorganism couldn't go. So by doing this experiment, he proved that over here living organisms were there. So that's the reason that allowed to grow other living cells. So, but here there was no living organism. So that's the reason life didn't grow over there. So this experiment actually disproved the spontaneous generation theory. Life cannot be grown from non-living material spontaneously. No, life only can grow from a life, or you can say the living cell. Now at this moment you can understand that we came to know the origin of cell. So all the scientists are behind the cell, okay. They really need to know how the origin, how cell actually do lots of things, okay. They were very curious that all organisms on earth have a cell. So all those questions were there. Sladen, he was a botanist. He used to work on plant. He actually showed that the plant tissue, one plant tissue actually divides into two or four. So basically plant tissue originates from plant tissue. So one cell divides into two and that's how they grow. Plant grows on that way, or number of plant cell increases because of the cell division. That's what first Sladen he told. Swan, he was a physiologist. He was working on animal tissue. He also observed the same thing, and then he discussed his opinion with Sladen, and finally these two scientists, they came in conclusion together that cell actually can arise from pre-existing living cells, and they proved that. Another scientist, Rimac, he also support that concept, like cell originate from other cell. Varco, he worked on cellular pathology. He told that all living organisms are made up with cell. So all together, all those scientists, there are few more scientists. Basically, all these scientists together, or you can say all the contribution of the scientists together, finally they proposed the cell theory. Cell theory, there are three tenets. Inside your book there are two, but actually there are three tenets, okay. So all living organisms are composed of one or more cells, okay. If it is a living organism, it must be made up of cell. It could be one cell, it could be multiple cell, but it is made up of cell. So that's the first tenet. Second one, cell is the basic unit of structure and organization in organisms, okay. If it is single-celled organism, okay, just if any organism, it has just only one cell, this one cell, just single cell, can do all the basic functions of life, okay. It can make the food, it can produce energy, it must have sensitivity, it can reproduce. So all the characteristic features of a living organism can be fulfilled by just one single individual cell, okay. So when when the organism is multicellular, all these cells together, whatever they do for to keep that organism alive, just one single cell is capable of doing all those things. Okay, so that's the tenet of second cell theory. Third one, cell arise from the pre-existing cells. So I told you, like all the scientists, they disproved the spontaneous generation theory. So cell has to originate from pre-existing cells, not from a non-living organism. This slide is very, very important. Now, endosymbiotic theory. So we are actually improving, you know, like we discovered the microscope. Okay, before that we had idea. We used microbes to ferment our food. We made our wine. We made cheese. We didn't know, but still we used microbes. Then finally we made microscope, and we identified that microbes, the presence of microbial world. Okay, we came to know about them. Then finally we identified that all those microbes are made up of cell. Then we identified that when it is a cell, even it's a single cell organism, means it is living organism. That we are there at this moment. We are there. We know even a single cell organism is a living creature. It can do, it can breathe, it can utilize energy, it can sense, it can reproduce, everything. Whatever we, you know, human, we are doing with trillions of cells, just single cell bacteria, microbe, they can do same thing. Whatever we are doing, they can do everything what we do. Now it actually came to our mind that, okay, so what's the difference? You know, like single cell bacteria versus we are multicellular organism. We have brain and we are walking. We can kill if we want. We can easily kill the bacteria. So what's the difference? Or how could we are so different? Okay, and how it became possible actually. So we are there. That how we originated. Basically, at this moment, we are not interested about the cell. We are interested about our origination, or you can say the life. We are interested about life. We are thinking about the life. Okay, so when we are thinking about life, how life originated. Okay, so one single cell versus multiple cell. It's easy to imagine that positively single-celled organism came before us, right? How we see that we are developing, you know, it step by step. So they imagine that positively the single-celled creatures, they came even before us. Okay, so they are our ancestor. So all the scientists actually accepted that, and we have scientific proofs also on that. So how we originated, okay, and or what happens so that multicellular organisms came out, or how multicellular organism came out from a single cell organism. Okay, so from there we started working on this endosymbiotic theory. So endosymbiotic theory tells us, actually it's a proof that all cells, actually we all were at the beginning, we were prokaryotic cells. Okay, what happened? All these cells, prokaryotic cells, accidentally. So and at the beginning, millions of years ago, all these single-celled organisms were aquatic, right? There was no land at that time. We are talking about that time when the earth was full of the ocean and water. There was no land. So what happened? All the food stuff, or you can say oxygen, carbon dioxide, whatever the minerals, whatever they need, it used to enter inside the cell by the process of diffusion, or you can say, or maybe osmosis. These are the two processes were there. So that's how they used to engulf, or they used to get the food. Accidentally, what happened? One prokaryotic cells actually engulf another prokaryotic cells. So one prokaryotic cell is inside another prokaryotic cell. And let's say this prokaryotic cell was aerobic. It used to do aerobic respiration. Okay, means aerobic respiration means it used to use oxygen like us. We do aerobic respiration, right? We inhale oxygen, we produce carbon dioxide. So all these cells used to do that. And when one large cell actually grabbed a small aerobic cell, single-celled organism, after a certain period of time, these two organisms, like this one and this one, the large one, they started doing symbiotic relationship. Symbiosis means it's understanding, you can say, with the biological communities, you can say. So basically over here, like let's say this is the aerobic bacteria and this is anaerobic bacteria. So this bacteria needs carbon dioxide and this bacteria produces carbon dioxide. So what happened? So these bacteria used to use carbon dioxide that actually helped the large one to get carbon dioxide easily. And when these bacteria used to eat, and after eating, after the metabolism or after the digestion, it used to produce oxygen, and these bacteria used to get oxygen very easily over there. Okay, so that's how these two organisms started symbiotic relationship. They got dependent on each other. Slowly, this aerobic bacteria actually converted into mitochondria. Like how mitochondria inside our body, we actually use oxygen. Okay, and that's how inside our mitochondria we produce carbon dioxide. That's our powerhouse, you can say, where we produce energy. Okay, now we break down C6H12O6 glucose in the presence of oxygen. Okay, and we produce carbon dioxide, we produce a high amount of ATP inside the mitochondria. So basically this mitochondria are nothing but prehistoric aerobic bacteria. Okay, they accidentally entered inside a large non-aerobic bacteria, and ultimately they started a symbiotic relationship, and slowly they get modified, and ultimately this aerobic bacteria converted into a mitochondria. And that is the endosymbiotic theory. We have another symbiotic theory about chloroplast or plastid. Exactly same way, instead of aerobic bacteria, large prokaryotic bacteria, if they actually engulf a photosynthetic cyanobacteria. Okay, and you all know that that chloroplast or plastid, they help plant to do photosynthesis, right? So it's same concept. So that's the endosymbiotic theory. So basically what happened at the beginning, that prokaryotic cells, they did not have any nucleus, they did not have any membrane bound organelles. Okay, but over here, after a long period of time, what happened? This bacteria converted into mitochondria, and it has membrane. So basically this cell, actually prokaryotic cell, converted into one eukaryotic cells, and that's actually describe the origin of eukaryotic cell. Okay, very interesting, and it has scientific proof. Like I hope you all know, like inside our cell we have mitochondria, right? And that's our powerhouse, where we actually breaks down the glucose, we produce ATP. So mitochondria, it's double membrane organelles. Okay, it looks like that. Double membrane organelles. It has its own DNA. It's organelles, but inside our cell, if this is our cell and we have our chromosome, we have our DNA, but this cell organelle, it has its own DNA. That actually clarify this endosymbiotic theory, because it was actually previously it was another bacteria. Accidentally they come inside another cell, large cell, and they started doing symbiotic relationship. So basically we do have symbiotic relationship with the mitochondria. So DNA, if we see the DNA structure of mitochondria, it does not go with our DNA. Okay, it does not resemble our eukaryotic DNA. It actually show the similarities with the prokaryotic DNA, bacterial DNA. Okay, so and another thing is and same inside the chloroplast, same thing. The other thing is this mitochondrial DNA. Usually, how we divide our cell, we go by the mitosis, right? From one cell, we produce two cells. And when cell division occurs, before cell division, what happens? We have one set of chromosome. We actually divide the chromosome into two sets, and then we distribute. One set of chromosome goes here, one set of chromosome goes there, right? That's the cell division process. That's the mitosis. That's what we do for our main DNA. But what inside our body, inside our cell, this mitochondrial DNA, how they divide? They divide by the binary fission, means how actually bacteria divides. Okay, so all these example, this is actually nothing but the scientific proof of endosymbiotic theory. Okay, so now we know that we all originated from a single prokaryotic cells, and because of this incident, just accidental incident, large organisms started eating the small organism, just like large fish eat small fish, exactly same way. And finally, they started working on the symbiotic relationship, and finally it gives a new group of cell, the origin of new group of cell has begun, and this group of cells called eukaryotic cells. What's the time? Do we have time? Yeah, we do have time. Okay. So now we know our origin and all those things. So we are moving to the germ again. Okay, how to control germ. So previously, like lots of people, like at this situation, you can easily understand that it's a new virus, that corona. Okay, completely new virus. It has similarities with the SARS, so we know that it's a group of SARS virus. It's a virus. We know the virus, how virus works or how virus behaves. We know all these things, but still we really do not have anything so that we can control or we can kill this corona. Okay, at this age with high technology and all those things, we are using bacteria, yeast to make medicine. But just imagine, we cannot control this virus. Okay, we are just working on it. Almost, it is going to be almost 1 year. Still, the whole world is behind this virus, small, minute creatures. Still, it's so difficult. So many people died and all those. Okay, now imagine. So it's 2020, year 2020. Imagine when it was 1818. Okay, they just had a very basic preliminary microscope. Okay, that IT was not that superficial. High technology was not there, and all those. So those people, they were struggling. They had idea, just like we have idea. It's a virus. We all know that's coronavirus. Okay, but we cannot control. Exactly same way. They knew tuberculosis. They know it's anthrax. It's plague. They couldn't control. Just like us. Like maybe after 200 years, the people will laugh at us that, okay, so the coronavirus, it's like after 200 years, it would be like influenza virus. But just try to imagine that period, 1818. So people used to die with cholera. Okay, the village after village, the town after town used to destroyed by all these diseases, like cholera-like disease. Now cholera, nobody dies at this moment. Okay, right? And lots of people even does not know the name, the disease cholera. Okay, so after 200 years, no, people will laugh that people died with corona. Exactly same thing. So anyway, so all these scientists, they were looking for how to work with all these germs. So Semmelweis, or you can say how to minimize the effects of these germs, and that's how we started making that germ theory of the disease. Okay. Semmelweis, he actually noticed that hand washing, okay, like now coronavirus, every time we are using antibacterial hand sanitizer and all those. So that identifying this thing, that if you wash your hand before treating patient or before doing the surgery, that actually minimize the death of the patient. It was big thing that time. So Semmelweis, he actually talked about that hand washing. Basically, they started recognizing the power of sanitization or all those things. Joseph Lister, he was a doctor physician. Before surgery, he noticed that lots of patient, most of the patient actually used to die after surgery because of the infection. So he discovered carbolic acid. He started using that phenol before and after surgery to reduce the infection, okay, contamination. Robert Koch, he gave us 4 postulates, so that still we use that how to identify the disease, okay, how to identify the pathogen, and how we can, by seeing the signs and symptoms, we can actually identify or discover the the disease. Oh, it's 7 o'clock. Okay, so let's keep today up to this. Okay, Monday I will start from this slide. Okay.
Have a good night.
Yeah, do you have any question?
No.
No, ma'am.
Thank you so much.
Okay, if you have any question, you can email me. So today up to this. Okay,
Have a good night.
good night. Yeah.